So these notes today are going to be about cell communication. So cells signal and communicate through direct contact, which are called gap junctions, secreting local regulators, which are growth factors and neurotransmitters, and then long distance messages, which are hormones. The cell uses all of these kind of at the same time, depending on what they're needed for. So which one of these gets used really kind of depends on what the cell needs at the time or what it's going to be used for. So an example here is showing you the cell-to-cell junctions, the gap junctions. Here is the cell communicating kind of in between two cells. These are usually the cells that are in the same tissue. So this will be two liver cells, two skin cells. And this allows them to communicate very quickly. Another thing you're going to see is this cell-to-cell recognition. So both recognition of self, meaning making sure the cell understands that it is a part of, the body, it's supposed to be there, so the immune system won't attack it. And also just to kind of say, hey, you're a liver cell, I'm a liver cell, let's do the same job. This is showing you the difference between long distance and local signaling. Obviously, local signaling is going to happen quicker because they're closer together. Long distance signaling is going to take longer. The advantage being local signaling is going to get the job done quickly, but the effect or the signal is going to go away quickly as well. So this is for something that needs to be done quickly but doesn't need to last long. For long distance signaling, this is going to take longer to get there, the signal, but it lasts for a longer time. It also takes longer to go away. So you're going to see a lot of neurotransmitter signaling here. You can see how close they are here, right? This is going to be nerve cells, local regulator factors, things that need to happen very quickly. Again, they're local because they're right next to each other. So the result of this, the effect of this signal is going to happen very quickly. Again, it's going to go away quickly. For long distance signaling, this is going to be your hormones. They're generally produced kind of at the top or bottom of your body, and they travel through your blood until they get to what we call a target cell. The target cell is the cell that is supposed to be acted upon. And again, because they have to travel through your blood and they're more long distance or further away, it's going to take longer to happen, but the advantage is it's going to stay around longer. And again, which one the cell your body uses really depends on what it needs. So there's three main stages to cell signaling. We're going to think of it like picking up a telephone. So the first one's going to be reception. That's basically picking up the phone and saying hello. This is detection of the signal molecule. We're going to call, from now on, not a signal molecule, we're going to call it a ligand. Ligand is the signal. So we're going to say, instead of signal, we're going to say ligand. The second would be transduction, or think of it almost like translation, kind of listening to what the person on the phone is saying and figuring out what they want. So this is conversion of a signal to a form that can bring about a cellular response. And then the response is the action. What does the cell need to do? That's the action that happens at the end of the cycle. So again, ligand needs to be in your vocabulary. Ligand is a small molecule that transmits a signal. So a ligand is a signal molecule. It binds to what we call a protein receptor on the outside of the cell. The way that this works, because remember with hormones, they're traveling through your blood, they're going to go and hit all kinds of organs. The reason why something, for example, like estrogen is only going to work on your ovaries is because your ovaries are the only ones that have the correct receptor. So if the cell does not have the correct receptor, it won't recognize the ligand. The ligand won't bind with it. That signal will not go to that particular cell. You can have intracellular or extracellular receptors. Intra meaning on the inside of the cell, extra meaning on the outside of the cell. And again, it depends on what it's used for. For the most part, intracellular receptors inside are nonpolar, extracellular are polar, which that should make sense because of that polar, nonpolar interior of the membrane. So again, this is just showing you the ligand. Here is the protein. Here's kind of the carb receptor. And then there is the ligand there kind of into the target, into the receptor cell. So this is where it's kind of connecting. This would be an extracellular on the outside. And then here's an intracellular on the inside. So this is showing you signal transduction just in picture form. It's the same three things every time, right? You have the reception, which is the ligand going into the receptor. You have the transduction, which is the processing and kind of converting of the signal to a useful form. And then you have the response, which is the action or the cellular process that needs to occur. So breaking it down even further, here we have an extracellular target cell. Here's the receptor. Here's the ligand coming in. So reception is that ligand connecting with the receptor on that target cell. The next step would be the transduction. So you see the relay molecule or passing the signal on or putting it in a useful form that the cell can understand. And finally, you have the cellular response, which is going to be the action that the cell needs to take, whatever that ligand told it to do. So looking at these in a little bit more detail, reception again is the picking up the phone, the hello. This is where the ligand and the receptor bind. You can again have a plasma membrane receptor, which is going to be an extracellular, that's going to be on the outside of the cell, or intracellular, that's usually going to be on the inside of the cell. These are going to be hydrophobic and small. So again, this could be testosterone or nitric acid can go inside the cell and do the shape like that. We're going to say that the ligand and receptor is highly specific. So that's what we were talking about before, where if the cell does not have the right receptor for the right ligand, it won't read the ligand, essentially. The ligand will just pass right by and it won't have any interaction with that cell. So the ligand's going to bind to the receptor protein. The protein's going to change shape, and that's going to initiate the signal transduction. That's kind of how that process is started. So there are 3 specific plasma membrane receptors that AP likes you to know. That's where I got these. I'm not doing this to torture you or just picked them out of thin air. So the first one's called a G protein couple receptor, or GPCR. The next is tyrosine kinase. And the third is a ligand-gated ion channel. So where you will see these could be a free response question where it'll say, what is the difference between an intracellular and extracellular receptor? Give an example of each. Give a specific example of each. Or give 2 examples of an extracellular plasma membrane receptor. So that's the level that you need to know this. We're going to go through each one. So the first one is the GPCR, or the G protein couple receptor. One of the unique things about the GPCR is how big it is. So you see it's actually made up of 7 proteins. I want you to think 777 when you think the GPCR. This is it, picture form, and then here's kind of the real form. Remember, proteins with that alpha helix. So the way it works is it kind of uses a relay protein, a secondary messenger. So you have the receptor here on the membrane. You have the enzyme that is going to be kind of what is acted upon. That's kind of where the response needs to happen. And then you have this G protein inactive. This is going to be the relay molecule. So what happens is you get the signaling molecule, or the ligand, in the receptor. It changes the receptor shape and activates it. That then activates the G protein by turning it from GDP to GTP. Think of it like it almost electrifies it. Then that GTP is going to move from the receptor to the enzyme that it's acting on. This is where it's doing the relay. It's bringing the signal from one place to another. And that's going to go ahead and activate the enzyme, cause the cellular response, and then the GDP inactivates and goes back and waits for the next signal. For tyrosine kinase, I want you to think 2. Tyrosine is 2, 2, 2, 2, 2. There are 2 receptors that need 2 ligands to start it. That's where we're going to do the TT222. So you have a tyrosine receptor here and a tyrosine receptor here. 2 ligands are going to go ahead and bind. And as soon as that happens, the 2 tyrosine kinase receptors snap together. That's kind of the movement that happens, the shape change. Once they do that, they're now called a dimer. You're going to think di meaning 2. This dimer is then going to activate kind of another kind of position in the transduction where 6 ATPs are going to be broken down. And you notice each one of the tyrosines gets a phosphate. This is called phosphorylation. This is adding a phosphate to the tyrosines. And then the cool thing that happens is they then go to the cellular response. And like you said before, you're going to think 2, 2, 2. So not only does tyrosine start with 2 receptors, 2 ligands, it does 2 cellular responses in response to that. And then finally, the ligand-gated ion channel you should be most familiar with. This is kind of the gated channel that we talked about before. This is one of those protein channels that's going to let things through the membrane, but it's gated, meaning it needs to be opened or shut. So what happens is you have ions that need to get through. The gate is closed. The ligand signal molecule comes in, fits in the receptor, opens up the gate. The ions will move through. That's part of the cellular response. This could be the K+, the sodium-potassium pump. Notice they're moving through normal diffusion, right? High to low concentration. And then once they're done moving, the ligand will leave and the gate will reshut. This slide here is just summarizing the 3 receptors, right? You want to remember G protein's really big. It has 7 proteins. It's going to use that G protein as a signal molecule. Tyrosine kinase, you're going to think 2, 2, 2. 2 receptors, 2 ligands, 2 cellular responses. And then the ligand-gated ion channel is going to be specific to moving ions in and out of the cell, including calcium and sodium. The next step in the process is going to be transduction. You can kind of think of this as translation. It's changing the signal into a form the cell can use and understand. You're going to hear a lot of cascading. This is where you're kind of relaying the signals or passing the signals on from receptor to target, kind of like a game of telephone you used to play when you were younger. The only difference is the signal is translated well. You don't mess it up. Two things that help this process along are protein kinases. Those are enzymes that help activate proteins, kind of like turning on switches. And phosphorylation cascades that enhance and amplify signals, helping it get to its destination correctly. This is showing you a phosphorylation cascade. AP does kind of like this, so I would kind of make sure you know what you're looking at here. They like to draw this. All you're looking at here is almost like a game of telephone. So here's the signaling ligand coming in, here's the receptor. And instead of this kind of receptor or the signal going directly from where it is to the protein down here, this is where the cellular response is occurring, it kind of uses this almost like stairs, almost like a slinky down the stairs, and it's going from one to the other, kind of activating kinases as it goes. So this is called a cascade. So you notice it's activating the first kinase, right? It goes to here. This activates this one, kind of like the game of Mousetrap, if you're familiar with that. And at the very end, it's going to activate this protein, which causes a cellular response. So again, don't forget the point of the phosphorylation cascade is to amplify the signal, to make it better, to make it more clear, to make it stronger, so that the signal can go from all the way to the extracellular part of the cell down here to the activated protein. So secondary messengers are exactly what they sound like. They're small, non-protein molecules or ions that relay a signal inside a cell. So again, most extracellular proteins signal kind of where the ligand goes are on the outside of the cell. And a lot of times the cellular response has to happen inside the cell. So the secondary messenger is a thing that uses that the cell uses to get the signal from point A to point B. You see some examples here. So the G protein receptor, the G protein is actually a secondary messenger. It's relaying the signal from one place to the other. You also can have things like calcium ions that act as secondary messengers. They're small, usually things that can move around the cell quite easily. And what they're going to do is they're just going to help move the signal from kind of where the ligand attaches to where the cellular response needs to occur. So one of those secondary messengers is called CAMP. It's cyclical adenosine monophosphate. You don't have to know the whole term. CAMP is fine. CAMP, again, is a secondary messenger. You can see it here. Okay, so it's taking the signal and moving it down kind of more interior to the cell to where the protein that's going to elicit the cellular response is. It's one of those things, again, it's a game of telephone, a game of Mousetrap, where it's relaying the signal down, converting ATP to CAMP, and kind of making that protein kinase become activated. So CAMP, I would know as an example of a secondary messenger. That's kind of what I would have in my brain. And then the last stage of this is going to be the response, which is basically whatever cellular action needs to take place. It could be regulating protein synthesis, causing the cell to make a protein, to stop making a protein, to turn a gene on, to turn a gene off, to make the cytoplasm do something, regulate activity in the cytoplasm. There is a wide variety of what that cellular response could be, but that's kind of the whole point. The ligand is passing the signal through transduction. You can see the phosphorylation cascade here, and then eventually it's going to go and do whatever the cell needs to do according to that signal. So this process of, signal transduction is happening constantly in your body, awake, asleep. It's happening right now. Most of the time it happens seamlessly. You don't even really notice it because your body's kind of doing what it needs to do. There are some diseases that the cause basically is a problem in that pathway. Somewhere in the pathway, either with the, reception, transduction, or response, there's an issue. So some examples here are diabetes. Diabetes is an excess of blood glucose because you don't make enough insulin. So what's happening there is there's some kind of issue with the body's reception of glucose. So when you have glucose in your blood, your body's response to that should be to produce insulin, which helps get rid of the glucose. And there's an error there. We're going to talk about cholera in a second. Any autoimmune disease usually has to do with the signal transduction pathway because autoimmune means your body's not recognizing self and it's attacking its own cells. Cancer is quite simply just uncontrolled cell growth. So either the cell is not getting the signal to stop replicating or it's getting a signal to replicate when it shouldn't. Things like neurotoxins, poisons, pesticides can interfere with this, especially neurotransmitters. They can be blocked by those things. And then we use some of this pathway blockage with drugs like anesthetics, antihistamines, blood pressure meds, where we can stop signals from going when we don't want to. So, for example, if you have too much of a signal going that's causing too much of a cellular response, we can use a medicine to kind of block some of those signals. That's one way where we block pain signals. So, cholera is actually a disease. You get it by drinking contaminated water, and the water is usually contaminated with human feces. It's a bacteria. It goes into your intestines and it messes with your G protein, so that receptor that we just looked at. And what that does is it helps, the G protein is responsible for regulating your water and salt levels. So when you have cholera, your G protein gets stuck in the active form and the intestines secrete salts and water. And basically, you develop profuse diarrhea until you die. So you're basically producing too much feces, too much water in your feces, and you're losing the water. So your body's kind of getting depleted of water and you can die from dehydration. It was a bigger problem, it's a bigger problem in certain parts of the world. You can still get cholera here. The only thing is with cholera is we're able to treat it pretty quickly. We put you in the hospital, we give you an IV of fluids to kind of replace the fluids you're losing. We can use an antibiotic for the bacteria and you pretty much will survive. There's still places where it's pretty bad. Cholera was a big deal, like in kind of like medieval times. Cholera was one of those first diseases that we figured out we could use chlorinated water to clean. So if you had a well in a town that had cholera, you could put some chlorine in it and it would kill it. So that's kind of where cholera's kind of claim to fame comes from. But again, the whole issue is it's disrupting that signal transduction. Apoptosis is cell suicide. So if a cell can detect that there's something wrong with it, that there's something fatal, it's going to actually get dismantled and digested. Basically, it's going to destroy itself to not further its damage. This is triggered by a cascade, a signal transduction pathway. The protein capsase is the one that goes and says, hey, there's something wrong with you, you need to get rid of yourself. What this does is it protects cells from damage. It helps development and maintenance. This is what catches, bad DNA or something wrong with the organelles. It'll get rid of the cell immediately so it can't proliferate that issue. We do think this may be involved in some diseases like Parkinson's and Alzheimer's. What they both have in common is the brain cells are getting destroyed when they really shouldn't be. So one of the idea is that they're getting the signal, they're getting that capsase signal to apoptose when they shouldn't. If that's the case, it's actually a really easy fix. We just need to block that signal. So we are making a lot of grounds with curing Parkinson's and Alzheimer's. This is just showing you apoptosis of a human white blood cell. So the normal white blood cell is on the left. The right is the white blood cell undergoing apoptosis. You can see it's kind of shrinking and forming these like big blobs. And eventually it will get digested and eliminated. Apoptosis is also what gives you digits, gives you fingers. So at the very beginning of your development, this is actually a mouse, but it's going to be the same with humans. Your hands and feet are actually kind of club-like. And then what happens is the cells in between your fingers get messages to apoptose, and they kind of go away, and that's how you get your digits. Sometimes this can go wrong. That's how you get webbed fingers and toes, where you get skin in between your fingers and toes because they weren't given the signal to apoptose in time.